Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this particular lecture, I will be explaining you a fatty acid chain elongation and desaturation of a fatty acid. Now let's get into details. Once a fatty acid is synthesized by fatty acid synthase complex, a fatty acid which can be elongated if there is a requirement for the cell to do so. Like generally a fatty acid that is synthesized is of 16 carbon, that is palmitic acid, and a 16 carbon fatty acid, it can be elongated into 18 carbon fatty acid or a 20 carbon fatty acid or a 22 carbon fatty acid depending on the requirement of the cell, especially when a fatty acid that is made in the liver and sent to other tissues in the form of by loading it onto VLDL in the form of triacylglycerol. So a specific cell, according to their requirement, it can be elongated as per the need. So the elongation of fatty acid can go on in two subcellular organelles. One is a microsomal system of elongation and other is a mitochondrial system of elongation. So in the microsomal system of elongation, so the carbon donor of fatty acid that is elongated, it is malonyl-CoA. Now the malonyl-CoA, which has got 2 carbon, it can be used to elongate a fatty acid by 2 carbon more. Example here, if a fatty acid that is shown here as acyl-CoA, which is a general fatty acid, I will just write that as a 16 carbon palmityl-CoA, just for an example. And this 16 carbon palmityl-CoA, if it has to be elongated, it will be sent into endoplasmic reticulum. That's what is microsomal system of elongation, happens in the endoplasmic reticulum, to be specific, smooth endoplasmic reticulum. So where a malonyl-CoA is acting as a 2 carbon donor, and the very first enzyme which is condensing this acyl-CoA 16 carbon palmityl-CoA with malonyl-CoA is 3-keto-acyl-CoA synthase enzyme, where condensation happens, and that is and also there will be decarboxylation going on. So basically in a fatty acid synthesis, where I have explained you a series of reactions, a spiral of fatty acid synthase reaction, that is condensation and decarboxylation I was explaining, then the reduction, then dehydration and reduction. These are number 1, number 2, number 3 and number 4. 4 series of reaction will be incorporating 2 carbon from malonyl-CoA, because one of the carbon is released as carbon dioxide here. So those 2 carbon malonyl-CoA will be incorporated to elongate a fatty acid by 2 more carbon. That means a 16 carbon palmitic acid can be converted into 18 carbon stearic acid. Where exactly these 2 carbon from malonyl-CoA added? Always note that carbons will be added towards the carboxyl side of a fatty acid. So here is the number 1 carbon, number 2 carbon, like that in a acyl-CoA molecule. So 2 carbons will coming from malonyl-CoA will be added here, which I am boxing, right? The one that is in the box. These are the 2 carbons which are added on to the carboxyl carbon, and the methyl carbon is pushed on the other side. So condensation, decarboxylation, where carbon dioxide is released, 1 carbon from malonyl-CoA is released there. Then the reduction, there is use of NADPH plus, dehydration and reduction, 1 more NADPH used, and the fatty acid is elongated by 2 more carbon, making 18 carbon. 16 carbon fatty acid converted to 18 carbon fatty acid. So for every elongation of 2 carbon elongation, there will be use of 2 NADPH plus 2H plus molecule. That's how the elongation of fatty acid is done. Whereas in the microsomal elongation system, so although it is less active, the carbon donor in mitochondrial elongation is acetyl-CoA, and it is NADH dependent instead of NADPH dependent. Coming to desaturation of a fatty acid. Now the desaturation or unsaturation of a fatty acid, it is incorporation of a double bond to a saturated fatty acid that has been synthesized. Example, if a stearic acid is there, which is an 18 carbon fatty acid, if a cell wants to make it as a oleic acid, which is an 18 carbon single double bond fatty acid. So double bond can be incorporated in our cells. We have enzymes called desaturases, and these desaturases, they can introduce a double bond to a saturated fatty acid, and this introduction of double bond are basically referred as desaturation or unsaturation. It will be going on in the endoplasmic reticulum. And the desaturation process, it needs molecular oxygen, and it needs NADH and cytochrome B5. And the most common desaturation generally involves the placement of double bonds between 9th and 10th carbon atom. And other possible positions where desaturation can be done is between 4th and 5th carbon atom, 5th and 6th carbon atom, and 6th and 7th carbon atom. Basically 4th, 5th and 6th carbons. So we have an ability to insert double bond a carbon atom 9 and below. Any carbon atom, any double bonds that are seen are 10th and above carbon, like between 10 and 11 and 11, 12, like that, we don't have ability to do that. We don't have desaturases to insert double bond beyond 10th carbon atom. Okay, so that's our limitation. And here on the right hand side, a figure that is shown where a fatty acyl desaturase enzyme, which is just a general figure here, it is inserting a double bond using molecular oxygen, NADH and cytochrome B5, because NADH is providing that proton and keeping this cytochrome B5 reductase in a reduced condition, and then ferrous iron, which is oxidized into ferric iron here, so where a double bond is inserted into a saturated fatty acid to make it as a mono unsaturated fatty acid. Now, we can also synthesize a polyunsaturated fatty acid, although we have a limitation to introduce double bond beyond the 9th carbon atom. But only thing is, if we have a essential fatty acid that is already having a double bond beyond the 9th carbon atom, we can use essential fatty acid like linoleic acid, which has got double bond at 9th and 12th carbon. It is an 18 carbon fatty acid which has got two double bond, and double bonds are present at 9th carbon and 12th carbon. So note that 12th carbon we don't have ability to introduce double bond. That's why we are taking it in the diet. It's an essential fatty acid, linoleic acid. Once we get linoleic acid, essential fatty acid, or alpha linolenic acid, which is 18 carbon fatty acid having three double bonds, one is at 9th, other is at 12th, and the other is at 15th position. Note that 12th and 15th position we cannot introduce double bond. We don't have desaturases to do that. Plants can do it because plants have delta 12, delta 15 desaturases, right? So once we get essential fatty acids having double bond beyond the 9th carbon atom, we can simply use that fatty acid and elongate and desaturate within our capabilities, within our enzymes capability. How to do that? This slide is showing you that. So look at malonyl-CoA, which has got double bond at 9th and 12th carbon atom, 18 carbon. So delta 6 desaturase coming in, and this delta 6 desaturase, it is introducing a double bond between 6th and 7th carbon atom. So we have a double bond here which is newly introduced by our human enzymes, delta 6 desaturase. So we have this gamma linolyenyl-CoA having double bond at 6, 9 and 12 carbon atom. We can do that because we have delta 6 desaturase, and then conduct an elongation process using malonyl-CoA as a 2 carbon donor. So where is the elongation? Always remember 2 carbons are added to the carboxyl carbon. So here is the carboxyl carbon, 2 carbons are added. Because of this, a position will change. Position numbering, the carbon numbering will change here, because The 6th position which was there originally, now that will move to 8th position because we have inserted number 1 and number 2 carbon to the carboxyl side. Accordingly, it will move on to the left side towards the methyl end because 18 carbon is now converted to 20 carbon fatty acid. Now this 20 carbon fatty acid having 8, 11 and 14 position double bond. Now it is undergoing desaturation one more time by delta 5 desaturase. We do have that enzyme. So it will introduce double bond between 5th and 6th carbon, and we have 20 carbon now having double bond at 5th carbon, 8th carbon, 11th carbon and 14th carbon. This particular fatty acid that is synthesized just now, it is a arachidonic acid, and this is how linoleic acid, which is an essential fatty acid, can be elongated and desaturated into arachidonic acid in our body. And note that arachidonic acid has 20 carbon, it has 4 double bonds, one is at 5 and 8, other is at 11, 14. So we are able to do it using available desaturases, but the provided we should have taken linoleic acid or alpha linolenic acid to do such things. That's how we can elongate a fatty acid in our cells. Okay, so synthesis of polyunsaturated fatty acids will come from using within our body. We can do that using an essential fatty acids that are coming from food, generally from plant source, and also consumption of fish like fish oils. So fish oils contain 𝐒𝐼𝐾osapentaenoic acid, 𝗏𝑑ocoshexoenic acid, and this fish obtains these fatty acids by eating phytoplanktons, the plants that float in water. Okay, so humans, elongation and desaturation of linoleic acid makes arachidonic acid, and the arachidonic acid, it is a precursor for prostaglandins, thrombosins, and these are inflammatory mediators. These are pro-inflammatory molecules. Elongation of and desaturation of alpha linolenic acid will make 𝐼𝐾osapentaenoic acid, 𝗏𝑑ocoshexoenoic acid. They are precursor for eicosanoids, and these are anti-inflammatory, pro-resolving and cytoprotectants. So like this, a polyunsaturated fatty acids can be further elongated and desaturated according to our cellular needs. So that's about fatty acid elongation process going on in the microsomes, and elongation and desaturation process which is also going on in the endoplasmic reticulum. That's about it. I hope this video has helped you in understanding the small concept of elongation of a fatty acid and also desaturation of a fatty acid. Thank you.